Yeah, thank you for giving me the opportunity to present my work, and I thank the organizers for hosting this very nice workshop conference here. So I'm going to talk about targeting function networks with TMS. I mean, I don't really have to do a big introduction, as from the talks from yesterday. It's like pretty clear that we have both direct and indirect networks effect with TMS. So we're not like only hitting one specific spot, but the activity also spreads to different brain areas as well. And we would like to also use those information from network effects to also improve our targeting of our stimulation protocols. That we not only want to stimulate a specific anatomical spot, but also target a network. And up until now, we're like largely missing models to predict a possible network effect of TMS and how we could use them to improve targeting. And so the idea that we had in the study was like using two techniques. One is like a TMS electric field modeling, which also, as in TDCS, is like used a lot to predict the anatomical region that is stimulated with TMS. And on top of that, we also wanted to combine it with resting state functional MRI as a tool for an in vivo assessment of functional networks. And like kind of merging those two techniques to create like a functional targeting approach. So that were like the two goals of our study, to develop a principled approach that allows us to determine functional networks stimulated in an individual, not on a group level, but really for each participant. And then we developed this approach, and we wanted like to evaluate it on an area that is like largely targeted for depression, for example, or other disorders in like the broader dorsolateral prefrontal region. So our method, so this is like more or less basically a modeling study. We took anatomical and functional MRI data from the Human Connectome project, which is like like a big study ongoing where they collect, I think, very high resolution anatomical and functional data from like 500 subjects, and we took 25 of them. And our approach was then like to use the anatomical data to create a biophysical model to estimate the effect of TMS and the functional MRI to estimate the functional networks. So we created for each subject a finite element model where we distinguish between like the standard tissue types. And we also included DTI anisotropy to estimate the anisotropy in the conductivity tensor. And then for each subject, we simulated on a grid of 5 by 5 points around the DLPFC region, possibly stimulation, targets, and orientation. As we already heard in the talks yesterday, that orientation matters a lot for TMS. It's like mainly started at the motor cortex, where you see, oh, if you rotate the coil by 45 degrees, my effects completely disappear. But for the dorsolateral prefrontal cortex, orientation is often completely neglected, where they just fix the same orientation as has been proven optimal for motor cortex, and take the same one for the DLPFC as well. That might be not the optimal approach, as like the gyrification can vary quite a bit at DLPFC and has a lot of variability between subjects. So we wanted to basically screen the whole parameter space that is possible, both in location and orientation, see how that would possibly affect the functional networks engaged. And to estimate those functional networks, we took a seed-based functional connectivity approach, where we took the electric field that is induced by TMS, thresholded that to just take like the maximum area, so kind of as a, say, estimate on the anatomical region that we stimulate, and then took at each node of the electric field, took like the time series from the resting state signal and computed a weighted time series for this target region. And this weighted time series we correlated with every other time series in the brain to create a functional connectivity map. That is what the functional connectivity map looks like. So we used here for the experts, we used partial correlation. That's where you see also like strong and negative correlation. You see that here in that basically we got strong correlations where we had like, a strong electric field, but not everywhere. We also got like negative correlations, and we got also then, for example, high correlations at remote areas, kind of indicating those areas that are then functionally connected with the target area. And we already see that here also at the DLPFC coil orientation matters. We have an example where you stay at the same spot and we just rotate the coil by 90 degrees. So one have like anterior posterior current flow and one like a left to right. And you see like the functional connectivity maps is basically inversed. So you could argue if by rotating the coil at certain positions, you might end up targeting a completely different network. Then we had like all those like 300 maps for each participant, and kind of, you know, to create like a summary of all those, we used a clustering approach based on community detection, where we then sorted functional networks that were similar to each other and maximally dissimilar to each other, to the other ones. And it just create a full connectivity matrix by taking the spatial correlation between functional connectivity maps with each other, and we basically saw then largely bimodal structure of two large networks, and if you cluster them, you see like those two big networks emerging. And based on those two networks, we could then sort stimulation areas and orientation, which then, based on when you stimulate there and which networks you would fall into. And we basically saw for all subjects like two different zones of stimulation. One is like more medial anterior and like also going here a posterior region, and one is like a lateral frontal region. And what was very interesting, that there were like certain regions that had both networks overlapping, but depending on coil orientation, depending how you have your coil oriented, you would then like flip from one network into the other at a certain point. So those are the two main networks. If you like compare them like with networks in the literature, you have like one like frontal parietal network, and the other is like what is called like as a default mode network that have also been reported previously in the literature to be involved in DLPFC stimulation for TMS. Those networks and also were found like on the group level, averaging over all 25 subjects. Based on those network parcellation, we could then create like a stimulation zone based on the group level, where we then looked for which position like in an MNI space we would then stimulate like a third network, and we again saw like those two networks again, like one stimulation zone here and one big here, where you basically, no matter if you're always in this region, you would always end up in the same network. But interestingly, those two areas were kind of separated by a stimulation zone, depending on in which subject you are, on which coil orientation you have. You might end up stimulating a different network. And those zones, they also was like roughly corresponding to an area where we had a high coil orientation sensitivity. And then in the following, we did also like a second analysis where we then tried to reduce like those, I think like 4,500 spots, like in over all 25 subjects, and in like only 9 zones, which we then just ordered by anatomical location, kind of make this problem a bit more tractable. And then we basically wanted to see, like, for each of those zones, in which, like, previously known function networks, our TMS stimulation areas would fall in. And then we used, like, a parcellation approach that has been introduced by Thomas Yeo, where he, like, found, like, functional networks in over 1,000 subjects, such as, like, the default network, frontal parietal networks, motor somasensory and vision networks, and kind of want to characterize our TMS networks, like, within this framework. And so what we did then is, like, for each of those 9 zones that we found, we looked which networks, our TMS networks, which would they be most similar to the Yeo networks. So, for example, here that would be the default networks, and it would be very similar to the networks in those points 9, 8, 7, and 5, 3. So basically, if you would, like, stimulate here in those zones, we would basically mainly end up in the default network. And for this one is the frontal parietal, that would basically fall within, like, the anterior and lateral zones. So basically, those are 2 main networks, and you also have some other ventral attention networks as well, which might be involved to, like, a lesser extent. Yeah, and in the final analysis, we did somehow meta-analysis of all of different depression studies that were used and reported coordinates where they stimulated the DLPFC to target depression. And first you can see, I don't know if you can see that very well, first those targets extend a quite large area. So that's not only that they have one spot that they hit all the time, but actually it varies over an area about 4 or 5 centimeters, nearly, or maybe not that much. And then we did the same analysis for all those depression targets, and what we saw, like, again, like, a big variability in functional networks involved. So we again got, like, the main networks, the default and frontal parietal networks. Yeah, just to see, okay, we have, like, a big variability in the functional networks that we may be targeting with TMS to the DLPFC. And this might be then also related to variability in treatment outcomes of TMS towards depression. Now, to summarize, we created a new principled approach to determine functional networks with TMS. We found that the involved networks vary in a systematic manner with call orientation and location. We could postulate different network stimulation zones for DLPFC. We found that depression targets varied strongly in their network profiles. Yeah, and this approach might be a practical guide to target certain networks at the DLPFC, but also be applicable to other brain areas as well, or, like, other brain stimulation approaches as TDCS, for example. Yeah, there's also present this as an HBM poster on June 17th, and I also want to thank my co-authors, Mike Fox from Harvard Medical School and Cameron Craddock, Stan Colcomb, and my supervisor, Mike Milham from the Nathan Kline Institute. Thanks for your attention.